Thank you. Okay. Great. So yeah, thank you so much, Maya, for organizing. And I'm just going to give some brief discussion of multimodal imaging and highlight a few examples. So we have many rich techniques available that we can use to study brain function, and each one of these is becoming increasingly more powerful, but any single technology still provides us with only a partial view into the brain. And so the idea is that we can try to integrate across different modalities to gain a more complete understanding of individuals or disease. And this might include combining complementary functional imaging techniques like EEG and fMRI, or combining functional and structural data. We can also record dynamic measures of peripheral physiology during imaging as well to provide more insight into the state of the brain and body at any given time. And so for human non-invasive functional imaging, one prominent combination has been EEG and fMRI, and I'm not sure why part of this is not displaying. Oh no, I think my slides are, there was something lost in the translation to this system. So use your imagination to fill in that there's an MRI machine over there along with a slower bold signal and an EEG. So these have complementary spatial and temporal resolution and are also sensitive to different biophysical characteristics. And so for quite a while now, there's been a lot of active development in ways of acquiring the signals together at the same time, and also in methods for fusing EEG and fMRI for analysis, where the data is either acquired simultaneously or separately. And so examples I wanted to highlight, methods like ICA can be applied jointly over EEG and fMRI to infer large-scale activation patterns associated with millisecond-scale changes in evoked potentials. We can look at correlations between EEG and fMRI to try and localize seizure onset zones for planning surgeries and epilepsy. And it may also be possible to kind of learn information across these different modalities. So can we train machine learning models to reconstruct some fMRI information from EEG and vice versa? And my picture on that is not showing up. So another area I wanted to highlight is in, okay, I'm sorry, there's actually an entire set of missing slides here. I don't know how that happened. That is very strange. Okay, well, let's see. Yeah, there's about 3 slides missing. But I kind of remember what they are going to, to say, so I can tell you, oh, they're just out of order. Okay, I found them. So another area I was going to highlight where this can be useful is in studying dynamic neuromodulatory states, including changes in wakefulness over time. So, these are continuously varying as we're scanning somebody. They also closely influence our behavior and cognition. And so one thing we might do is use, for example, EEG or pupil diameter, which are established measures of arousal, as a readout of someone's arousal state. And then if we've simultaneously recorded fMRI, then we can look at the kind of large-scale signatures that accompany those state changes. And so overall, this might be useful for advancing basic understanding of these states, their potential disruption in disease, as well as modeling state-driven variability in individuals, which actually can be quite considerable. So just a couple examples from our work on this. It seems that there's pretty reliable signatures in fMRI of changes in alertness. We can see these in humans as well as also in macaque monkeys. And while EEG or pupil diameter are more well-established in terms of getting us alertness information, it seems that these patterns can even allow us to detect at any given moment how alert someone is during your fMRI scan. So we try to test this by seeing if we can predict somebody's upcoming response to a stimulus based on the expression of these patterns in the fMRI data at a given moment. And another thing that's come to light in doing these studies of arousal is that there's this really closely linked response between the brain and the body. So a collaborator, Taylor Bolt, he brought together 10 different data sets. These are multimodal data sets. Each one had fMRI as well as one or more measures of pupil diameter, heart rate, respiration, and so forth. It seems that there's this large-scale reproducible mode of brain-body covariation that you can find explaining a lot of variance in resting state, but also linked with kind of arousal events that might happen, spontaneously in sleep or might be triggered by a task. So we can kind of see these patterns in many contexts. So I also wanted to speak a little bit on using physiological effects signals together with imaging. So I think in fMRI anyway, we have this notion that physiological effects like heart rate and respiration, that these are artifacts. And there are some artifacts induced by these signals. There's changes in the magnetic field induced by breathing. But there's other features of this physiology that closely co-vary with bold responses. And if we record these together and we can look at things like, does the hemodynamic response in the brain to a change in breathing or heart rate tell us about the integrity of the brain's vascular responsiveness? And can we also use experiments like this one they have done in animals to try to isolate large-scale networks in the brain that are related to controlling your respiration, for example. And so I think that there's a lot of opportunities for using this also together with fMRI. And there's also movement, to try to facilitate this in the field. So PhysioPi, which is highlighted here, is a community effort to try to facilitate physiology together with fMRI. And so just going to the end now, I think more broadly, there are many avenues and challenges that are interesting in terms of multimodal functional imaging. I only, spoke on a couple of examples, but, we can try to advance our brain-behavior relationships, understand individual differences. We can also try to better understand our measurements. So like fMRI, for example, is an indirect measure of neural activity, but can we if we were to use, electrophysiology together with fMRI, can we get a clearer understanding of what we can and can't infer? There's open questions and challenges in improving acquisition of multimodal data, and also, how can we more completely leverage the advantages of different modalities? I know like sometimes in our work in EEG and fMRI, we kind of make a sacrifice in one of the modalities in order to match it to the other modality, but then we're kind of missing some information there. So I think that there'll be some interesting modeling work that can try to connect these on different spatial and temporal scales. So yeah, thank you so much.